Hello, my name is Pietro Cippà. I am the social media editor of Transplant International. Today it is my great pleasure to talk to Karl Hillebrandt. He's a young physician, scientist, working in the Department of Experimental Surgery at the Charité University in Berlin. Karl recently won the Rising Star video session organized by the young professionals in transplantation at the ESOT Congress in Brussels. As a part of this award, Karl will have the opportunity to present himself and his work in this video interview today. So, Karl, could you explain what was actually the topic of your video presentation?
Yeah, the topic was the optimized red liver decellularization through portal vein or hepatic artery under oscillating pressure conditions.
And what was the peculiarity of your technique compared to the technique of your competitors?
Yeah, thanks for this question. Yeah, when our group started decellularization back in 2011 and 12, there were only a handful of red liver decellularization protocols. And when we started our experiments, we first aimed to reproduce some of these protocols, but we recognized that the success rate in our lab was really low, and most of the livers looked really inhomogeneous while as well as after perfusion. So macroscopically, some regions were decellularized after a short period of time, and other regions stayed non-decellularized until the end of the procedure. We were asking ourselves, why is how can we optimize the homogeneity of decellularization? Because from our point of view, this is was the main problem of the decellularization protocols we tested. And then we made literature research, and this research presented a work where they showed the positive effects of respiration and oscillating pressure conditions on the microperfusion of the liver. And we then abstracted this hypothesis that with these oscillating pressure conditions, maybe we achieve a more homogeneous decellularization. And actually this worked really well.
Okay. You are working with a liver model. The perfusion of the liver is quite unique in the body, actually, with the portal system, the artery perfusion. Do you think that the same approach would be successful also in other organs where the portal system is actually not present?
Yeah, you're right. This is a really unique situation we have in the liver with the 4 possible inlets for perfusion. In detail, the hepatic artery, the portal vein, the bile duct, and the hepatic veins. I think the arterial perfusion, as we do this, is applicable for other organ systems, and it's well known for decellularization, for example, in the kidney. If oscillating pressure conditions have a positive effect on decellularization of other organs, they are still not investigated. From my point of view, I think it will not have a positive effect on other organs, because the liver has this unique position under the dome of the diaphragm where the microperfusion is instantly related to the respiration.
Mm-hmm. So if we come to the mechanism related to your technique, do you think that this positive impact of the oscillating pressure condition is related to a mechanical effect, or do you think that your particular technique induce a kind of biological response related to the particular stimulation of the vascular structures?
Yeah, that's a good point. In the decellularization setting, we think it's only the mechanical effect where the microperfusion is optimized by applying oscillating pressure conditions. But in an extracorporeal perfusion setting, I could imagine that there's a biological effect of the pressure changes, and but to my knowledge, this is not was not investigated until now. This was not. But this oscillating pressure conditions become more relevant now in our experimental setting where we perform recellularization, because we're actually investigating the effect of oscillating pressure conditions on the cell distribution, the cell survival, and the cell function while as well as after recellularization.
Okay, this would be another application of your approach, actually.
Yeah, definitely.
If we think in a more clinical way, probably the ex vivo perfusion of organs, of course, might be important in the future to create organ scaffolds, but at the moment the hot topic would be rather to try to optimize the perfusion of organ to increase the quality of the organ before transplantation. Yeah. Do you think that the same approach would be actually would have positive impact also in this particular situation?
These findings we made and other groups before us are, I think, really relevant for extracorporeal organ improvement. But in the actual scientific discussion about liver preconditioning and extracorporeal organ improvement by perfusion, surprisingly, this topic is not considered at all. And none of the commercial liver perfusion machines is actually using oscillating pressure conditions to optimize the microperfusion. But it will be interesting if there is any change in the future.
Yeah, that's good. One of the very important aspects related to a better decellularization of organ would be that a more homogeneous decellularization may actually reduce the immunogenicity of the graft. Did you test that in your model?
Yeah, that's a really hot topic, because up to now there is no universal definition of what is a good and what is a bad decellularization. And moreover, we don't know from which point in decellularization there is the reduced immunogenicity. And in an optimal setting, we would hope that after decellularization, all immunological materials are removed, while the extracellular matrix remains, and we think the extracellular matrix is non or less immunogenic, and we know that the extracellular matrix is highly preserved over the species, so we wouldn't expect any immunological.
So you would speculate that if you would be able to reconstitute the organ using cells derived from the patient, you would not need immunosuppression.
There I come to our experience. We are actually investigating this topic. We have defined different decellularization protocols, and with a gradual difference in the decellularization quality. And we then implanted some of these tissue samples in a rat rejection model. And we looked for immunorejections, but this is ongoing research and I will not go into detail.
That's very cool. Now coming to another perspective of the topic, you actually won this prize in this video session at the ESOT Congress. This is a quite new format of presenting research. How was your experience by presenting your data in a video format?
I like this format, and I really would like to see this kind of session in Barcelona in 2017. So for me the challenge of the session was that you have this defined video and you have to give your talk parallel, and you have to really be on time, that you stay synchronous to your video. But already the preparation of the presentation was really different, because you have your default time frame and you have to think of which step I have to show in detail and which step is not that necessary to get a good impression of the whole procedure. Furthermore, it was also important for me that somebody who's not in this topic at all can also follow the whole procedure. So I decided to support my video with some simplified graphics. For example, I showed the rat cetus, where somebody who had never operated a rat will not have any idea how it looks. And there we see the hepatoduodenal ligament, and there are a lot of vascular structures. And so I made a standing picture, and there I put in a small graphic so that anybody who's seeing this yeah, can think, okay, this is this structure, and he's doing that. So yeah, the goal I wanted to achieve is that the auditorium, yeah, can follow the whole video and gets a better understanding of what is done right now.
And if you would have to give one advice to the next person who would want to win this session at the ESOT Congress in Barcelona, what would you say?
Yeah, keep it simple and short and have nice images.
Okay, thank you very much.
Yeah, thanks.
Thank you very much, Karl. It was great to talk to you today. Thank you very much also.